Now, let us move to the next question. The technique employed to introduce only one or set of desirable genes without including undesirable genes into the target organism is, the options are tissue culture, option 2 hybridization, option 3 mutation breeding, option 4 genetic engineering. So, carefully look here, the technique employed to introduce only one or set of desirable gene, means very selective genes we want to put into the target organism, that is organism of interest. What do we call this technique? When you look into the option 1, tissue culture, I hope you know plant tissue culture, it is used for micro propagation of plants. So, we take a little of living tissue and we are going to culture this under a suitable nutrient medium under aseptic condition to obtain a plant. Remember, the father of plant tissue culture is Haberland. So, tissue culture is for micro propagation of plants, thereby that is not our answer. So, we are going to get similar, genetically similar type of plant, any number of plants we can get in a short duration of time. The second option is hybridization. We all know crossing of two pure breeding parental stocks differing in certain features is called as hybridization. Hybridization is one of the traditional technique used by man for animal and plant breeding with an intention of improving. And here entire genome of the two parental stock is transferred into the hybrid or the offspring. Thereby, along with the desirable, always there is a chance of undesirable genes also entering into the hybrid. So, even hybridization is not an answer. The third one is mutation breeding. Here we are using radiations or chemicals to induce the mutation, and then we are going to raise the plant and check for the desirable characters. Here there is going to be a change in the nucleotide sequence that is going to give rise to a new allele, and thereby even this is not an answer. So, now we come to the fourth, genetic engineering. Genetic engineering is nothing but recombinant DNA technology. The most important aspect of recombinant DNA technology or genetic engineering is we are going to identify the desired gene or a set of genes, and then using a suitable vector we transfer into the organism of interest, and thereby it is genetic engineering which is the correct option. Now, we move on to the next question. For the multiplication of any alien piece of DNA in an organism, it must be linked with. So, carefully look friends, alien piece of DNA in an organism, it must be linked with. So, for the multiplication, options are 1 antibiotic resistant gene, option 2 DNA sequence called ORI, that is origin of replication, option 3 RNA primer, option 4 both antibiotic resistance gene and ORI DNA. Many students are going to commit mistake if they have not understood the question here. So, thereby my advice to you is always read the question properly, understand important term based on which you have to look for an answer in option has to be underlined before you go for selection of option. Let me again read the question and then we are going to have a discussion to arrive at the answer. For the multiplication, so I underline this also, of any alien piece of DNA. Alien piece means the gene of interest, which we are going to put into the organism of interest, where it has to multiply so that my gene, what I have put there, has to express and I should get the product of that gene. So what is required? Antibiotic resistance gene. Remember, antibiotic resistant gene is going to be used as a gene marker to know whether the bacterium into which I have put the gene has taken it or not. So thereby this is not the one what can be an answer. It is used as a gene marker. DNA sequence called ORI. So remember, if I am going to put a piece of DNA into an organism, it is not going to express, instead it is going to get digested in the organism of interest. And if it has to replicate, multiplication is nothing but here replication or duplication, producing several copies. And then it is very important that we have to always attach a ORI site. You have to put into the chromosome, or I am going to use a vector with a ORI site. So thereby option 2 is the answer. Let us look into other also. RNA primer. I hope all of you have studied DNA replication, you remember it. Primer is a must for DNA polymerase to synthesize a complementary copy of DNA on a template. So during replication we talk about a oligonucleotide of RNA acting as a primer. So that is not relevant here and it is not an answer. Both antibiotic resistance gene and ORI DNA, as already we know, antibiotic resistance gene is not the one what is required for multiplication of alien piece of DNA, thereby even this is not our option. So answer is option 2. Please remember, carefully we have to look into it. After going through all the 4 options, we should know which has to be marked as the correct answer. Dear friends, now we move on to the next question. Which scientist was first to develop the method to isolate plasmid and insert them into the bacterium? Options are Herbert Boyer, Stanley Cohen, Hamilton Smith, Paul Burke, all wonderful scientists. So here the question is, the scientist associated with isolation of plasmids and inserting back into the bacterium. I hope all of you know, plasmids are extra chromosomal DNA found in the bacteria. Plasmids are used as vectors to transfer the gene of interest. And with this, when we look into the option, Herbert Boyer. Remember, Herbert Boyer's greatest contribution to the field of recombinant DNA technology is his working on restriction enzyme EcoR1 to cause a cut of the DNA with sticky or staggered ends. So Herbert Boyer is not an answer. Stanley Cohen. Stanley Cohen was the first to learn the technique, develop the technique to take the plasmid out of the bacteria and put it back into the bacterium. Of course, Stanley Cohen later collaborated with Herbert Boyer and they are the two get a recombinant DNA. First, successfully they put the recombinant DNA into a bacterial cell. So that's the reason when you look into the history of biotechnology, we classify the history into two periods, old biotechnology and a new biotechnology or modern biotechnology. In fact, it is Boyer and Cohen name is honored here. Old biotechnology is called as BBC and new biotechnology is called as ABC. What is this BBC and ABC? By this time you know, before Boyer and Cohen, ABC after Boyer and Cohen. So Cohen is the one who isolated, who developed the technique to take out the plasmid and put it back into the bacterium, thereby that is a correct option. And then we look into the other two options also. Hamilton Smith. Remember, Hamilton Smith was the first person to study and type the restriction enzyme, what we call as Hind2. Haemophilus influenzae is the bacterium from which this plasmid is, this restriction enzyme is isolated. Paul Burr is called as father of genetic engineering. He was the first person to get a recombinant DNA. I am not saying putting a recombinant DNA into an alien organism or into a bacterium. So Paul Burr, he used lambda virus DNA and simian virus DNA and he fused them together to get a recombinant DNA. So first person to get a recombinant DNA, that is man-made DNA, that credit goes to Paul Burr, because of which he is called as father of genetic engineering. Now we move on to the next question. Restriction enzymes are commonly called, very simple question but very important. Option 1, cloning vehicle. Option 2, cloning factories. Option 3, molecular scalpels. Option 4, molecular stickers. So I ask you to remember what all these four actually stand for. Of course, you can very easily pick the answer, I hope. Restriction enzymes, already we know Hamilton Smith was first to isolate the restriction enzyme. We called it Hind2 because it was extracted from Haemophilus influenzae. Remember, more than 900 restriction enzymes have been so far isolated from about 230 strains of bacteria. Remember, restriction enzymes are the digestive enzymes. They are the one which are going to be produced by the bacteria to protect themselves from the attack of bacteriophage, that is virus. We all know virus is an obligatory parasite and we are going to classify the virus into three groups: bacteriophages, phytophaginae, zoophaginae, and bacteria attacking virus are bacteriophages. So with this, now let us look into the option. Whenever we talk about recombinant DNA technology or genetic engineering, it has made possible because of some wonderful tools, and here the tools are mentioned. Cloning vehicle, I hope all of you know. Cloning vehicle, vehicle means the one which is going to carry a passenger from one place to another, to the destination. And here the vehicle that we are going to use is called vector. It may be a plasmid, it may be a phage, it may be cosmid, it may be phagemid or phasmid we call it. It can be bacterial artificial chromosome, yeast artificial chromosome, human artificial chromosome. So cloning vehicle is a vector that is going to be used in recombinant DNA technology to transfer the gene of interest into the organism of interest. Thereby, this is not the option. Cloning factories. Remember, genetic engineering is also called gene cloning. Here we are putting the desired gene of interest to get its product, where the organism as it multiplies, the cell where we have put it as it multiplies, your gene of interest also multiplies, which is called as gene cloning. So, cloning factory is nothing but the host cell. It may be a prokaryotic host or eukaryotic host. One of the most common prokaryotic hosts that we use is E. coli. When you talk about eukaryotic host, we use the yeast Saccharomyces also. So, even option 2 is not our answer. Now we come to option 3, molecular scalpels. Now dissections are banned, we do not dissect, but scalpels are going to be used in surgery. What for? To cut. Molecular scalpels are the one which are used to cut the DNA. To cut the DNA you require an enzyme. DNA is nothing but a polynucleotide chain, and for that the enzyme that is going to be used is nuclease. You have read in your 12th standard, when you talk about nuclease as the digestive enzyme that are going to digest the nucleic acid, we have classified the nuclease as exonucleases and endonucleases. Exo means from extreme end they are going to cause a cut in the phosphodiester bond present between adjacent nucleotides. Endonucleases are going to cause a cut in the interiors, and here the term that you have to look carefully is restriction enzymes. Restriction enzymes are abbreviated as REN. They are nothing but restriction endonucleases. Endo means the one which is going to cause a cut in the interior. Restriction means at a specific site, we call that as restriction site. So, it is commonly referred to as chemical knife, molecular scissor, molecular scalpel. Thereby option 3 is the right answer. Now let us come to the 4th option, molecular sticker. I hope all of you know molecular sticker, also called as molecular sutures, also called chemical glues. In recombinant DNA technology, we are going to attach a piece of foreign DNA carrying the gene of interest with a vector DNA, and they have to be attached to make a hybrid DNA, a chimeric DNA, that is recombinant DNA. To fix the two, we require a glue. The enzyme that serves as a glue is DNA ligase. DNA ligase in recombinant DNA technology commonly referred because of its property of attaching the DNA of two sources as molecular sutures, molecular stickers, chemical glues. So, this is DNA ligase. So, this is not our option. The molecular scalpel is the restriction enzyme. Now we move on to the next question. The DNA sequences showing two-fold symmetry or reverse repeats are called options: pangenes, option 2 palindromes, option 3 plasmids, option 4 cosmids. So, remember here the DNA sequence showing two-fold symmetry or reverse repeats are called bifold symmetry, I call it. Pangenes. Pangene term is used when we talk about evolution. In fact, Charles Darwin, who had a very less knowledge on heredity, he proposed a theory to explain inheritance of characters, what you call heredity. He proposed the theory of pangenesis or pangenes. And when we talk about this, what this theory says, it says that the information from every part of the body is going to be carried in the form of discrete units called gemmules or pangenes through the medium of blood, brought to the gonad, put into the gamete, and that's how the characters are going to be passed from the parent organism to offspring. And this theory is totally discarded, but nothing to relate with our question. So, this is not a right option. Second option, we come to palindromes. I hope all of you heard of the words like Malayalam, Madam, Dad. What is so special about this? Here if you look into the spelling of the word, from both the sides, they spell the same, M-A-L-A-Y-A-L-A-M, Malayalam, M-A-L-A-Y-A-L-A-M, Malayalam. Madam, M-A-D-A-M, M-A-D-A-M, Dad, D-I-D, Dad, from both the sides. We call such words as palindromes. If they spell the same from both the direction, you call them palindromes. In case of DNA, we know that DNA is a two-stranded polynucleotide chain having a orientation of 5' to 3', and the two strands are anti-parallel. And if you are going to read certain sequence, if they are going to be same in the same orientation in opposite direction as they are anti-parallel, we call such sequences as palindromes. Here, when you look into the question, the DNA sequences showing two-fold symmetry or reverse repeats. For example, if you take EcoR1, I hope all of you know restriction enzyme, it is going to identify specific sequence of nucleotides in the DNA molecule, and it identifies, say, from 5' to 3' orientation. The two strands are of opposite orientation. It identifies the 6 nucleotide sequences: G, A, T, T, C. And the two strands are complementary. Thereby, naturally, when we look into the nucleotide of the other strand, G pairs with C by 3 weak hydrogen bond, A pairs with T by 2 weak hydrogen bond, again T 2 weak hydrogen bond, A 2 weak hydrogen bond, A 2 weak hydrogen bond, G 3 weak hydrogen bond. So, if you look here, read it from 5' to 3', G A A T T C, opposite direction, again 5' to 3', G A A T T C, and we call this as palindromes, and such sequences are going to be identified by restriction enzymes. In case of EcoR1, it identifies this, and it is going to cause a cut by breaking phosphodiester bond between G and A. And now here you are going to have thousands of nucleotides, this side you are going to have thousands of nucleotides, and they are going to be held by only this part. The strain is going to split them, giving staggered end, and that is the reason why restriction enzymes are called as molecular scissors. So, option palindrome is the answer here. Now, let us look into the other plasmids and cosmids. Please remember, plasmids and cosmids are nothing but hybrid DNA vectors. In case of plasmid, we are going to use the ori site of the phage and plasmid. In case of cosmid, here we are going to use cos site of the plasmid. So, these are nothing but DNA vectors. This is nothing but the Darwin's theory, where he says that in the form of pangenes or gemmules, the hereditary characters are going to be transferred from the parent organism to the offspring. So, answer is palindrome. Okay, we come to the next question now. Gel electrophoresis is used for option 1 DNA fragmentation, option 2 DNA fragments separation, option 3 DNA fragments fusion, option 4 DNA transfer to nylon membrane. So, one by one we look into it. Electrophoresis, I hope all of you know, you have studied gel electrophoresis. If I have to tell you specific, agarose gel electrophoresis. Remember, it is a separation technique, finish. So, thereby when you look, here we are going to feed the DNA. DNA is negatively charged molecule. The DNA that is going to be fed is in the form of fragments. Before that, we have taken the DNA, exposed it to restriction enzymes for digestion. We get the DNA of different length or molecular weight, and we have to separate, and for that we are going to use agarose gel bed, and as it is negatively charged, DNA is made to pass towards the anode. On the basis of molecular weight, they start moving, and over a course of their movement they are going to separate. Then we have to use a dye, stain them, observe under UV light to pick the DNA bands. And remember, this is also done in case of DNA fingerprinting. So, with this we look into the options. DNA fragmentation. DNA fragmentation is nothing but DNA digestion, and for this we are going to use restriction endonuclease enzymes or restriction enzyme. Thereby this is not our option. This option is not correct. Second, DNA fragments separation. Yes, this is our answer. Option 2 is the answer. Here we are going to separate the DNA fragments on the basis of their molecular weight. Smaller the length of DNA, lesser is the molecular weight, greater the distance it is going to cover in that duration of time. The third option, DNA fragments fusion, which already we know for fusing the DNA obtained from two sources. As we know, one is the gene of interest, second one is vector DNA. We use the enzyme DNA ligase. So, this is not our answer. DNA transferred to nylon membrane. Normally this is going to be done in case of DNA fingerprinting that you have studied in molecular basis of inheritance. And the technique of transfer of denatured DNA fragments, that is single stranded DNA fragments, from gel bed to nylon or nitrocellulose membrane is called as southern blotting. So, even this is not an answer. So, thereby the answer is option 2. We move on to the next question now. In rDNA technology, that is in recombinant DNA technology, the plasmid vector must be cleaved by, cut by. Let us come to options. Option 1, specific restriction enzyme. Option 2, same REN, that is restriction enzyme or endonuclease that cleaves the donor DNA. Option 3, DNA ligase. Option 4, DNA polymerase. Let us discuss this. Recombinant DNA technology, plasmid vector must be cleaved by. We all know that plasmid is used as a cloning vehicle. We have to open it and we have to put the foreign DNA piece, alien DNA, and then we attach to get a recombinant DNA and we pass it on to the organism, that is bacterial cell. And how to attach the open plasmid with foreign DNA fragment representing the gene of interest. Then remember, we are going to match the single stranded extension, what we call sticky ends, staggered ends. For that reason, the restriction enzyme what is going to be used to obtain a fragment of foreign DNA with sticky end, the same kind of restriction enzyme also has to be used in case of plasmid. So, when we look here, specific restriction enzyme, same restriction enzyme that cleaves the donor DNA, option 2 is 100% correct. That is the answer. We have to be very careful. The moment we come to specific restriction enzyme, yes, we do use. Which specific means? Then remember, it should be common, because we have to have complementary matching so that we can attach them using DNA ligase. When you come to option 3, I hope all of you know, DNA ligase is used to attach the two DNA fragments to get a recombinant DNA. DNA polymerase is a replication enzyme. So, thereby 1 is not our answer, 3 is not our answer, 4 is not our answer. The second option is the correct answer.